Welcome to the Mandel Show. I am your host, Mandel, and tonight we are learning about the mother, Anna, and her troubles trying to find the father of her baby. Please welcome Anna to the stage. Now, Anna, can you tell us your story?
Yeah, so I am a single mother and my baby is 7 months old and I don't know who the father is. And there are 5 possible guys and I decided, I don't want my baby to grow up not knowing her father. So I thought I'd come on this show and try and figure out who it is.
Wow.
Now, let's meet the fathers. This is Fletcher.
No, it's not my baby.
This is Oscar.
Sir, you're on camera now.
What? The baby can't be mine. I was in Europe doing a radical bike race at a time.
This is Jacques.
That is not my baby.
This is Bill.
It can't be mine. I use protection.
This is Clark.
It could be mine. I like children.
So, Anna, who do you think the father could be?
I have a feeling it might be Fletcher. The timing would be right.
Let's find out!
We went to a genetics laboratory where they tested the DNA.
Before we discuss the DNA testing process, we will discuss restriction enzymes. Restriction enzymes are made to recognize specific sequences of DNA and cut them in specific spots. So, for example, a restriction enzyme on this DNA strand would move in one direction and cut in specific spots where it recognized a specific sequence. In this sequence of DNA, a restriction enzyme may recognize the sequence CCC, GGG, and on the other side GGG, CCC, and maybe it'll cut right down the middle. Or maybe a different restriction enzyme would recognize this sequence of DNA and it might cut between the C and the A on those strands. Another restriction enzyme might recognize this sequence and maybe it'll cut between the T and the G.
Hello. My name is Samuel, and this is our DNA lab. So for this process, we had to add the 5 DNA samples from each of the fathers, or possible fathers, and 1 DNA sample from the baby. We added a restriction enzyme to each of the capsules, and that cuts up the DNA into varying strands. Because the father and baby's DNA is very similar, they will have similar strands, which means we'll be able to find out which father or possible father is the actual father.
Okay, and these are the results we got. We substituted the father's names with father number 1, 2, 3, etc. as not to give away the results. But basically, we got various marks on here depending on how long the DNA strands were. As you can see, the baby and father number 3 have very similar length strands, and that would indicate that father number 3 is related to the baby. But we did make some errors. As you can see, there are no marks under father number 2, and that's because when we were trying to inject the DNA, we poked through the gel and none of the DNA actually went into the well. So father number 2 could theoretically be related, but because the baby and father number 3 are so similar in their DNA, it's pretty safe to assume that it's father 3 that's related to the baby.
In my hand, I have the DNA tests to find out who the father is. Bill, you are not the father.
I knew it was mine.
Clark, you are not the father. Fletcher, you are not the father. Yay. It comes down to 2 possible fathers, Jacques and Oscar. Oscar, you are not the father.
Sick! I knew Mountain Dew would save me.
Congratulations, Jacques.